This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 110, recorded on August 13th, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
I'm well. How are you?
I'm doing fine.
Are you still in a water drought situation in California?
We are in a perennial water drought, but there is all sorts of talks about a very powerful El Nino coming our way in the fall. And I don't know, there's going to be a lot of water. I don't know if it's going to do any good towards relieving the drought, though, because that's more complicated. It has to snow in the mountains for it to do some good. Anyhow, we're waiting.
It's too bad. We hear all about it out here. We have lots of water, so I wish we could give you some of ours. Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello, everybody.
How are you?
Great.
Having a great summer?
I sure am. It's just too darn fast.
It's almost over, isn't it?
It's making me nervous.
When do classes begin at Michigan?
I don't teach in the fall, so I don't keep track. But we start our football season.
Yeah, that's what matters.
On September 3rd in Utah.
Oh, wow. In Utah. Are you going?
I am.
Wow.
You're going to Utah?
You bet. It's
Wow.
Jim Harbaugh's debut. And it's Memorial Day weekend. So then afterward, we're going to go on a raft trip on the Colorado. I'm hoping there's still water there.
Wow, that's pretty impressive.
Yeah.
Also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How have you been?
I've been good. I was up at ASM headquarters last week talking to the Futuring Committee about branches and learning the great plans for ASM's future. So it was a rather fascinating opportunity to talk with Lynn Enquist and Tim Donahue and Victor Rita and all those folks.
Yep. Yeah, always nice to go to ASM headquarters. It's fun. I will be in Washington tomorrow.
What's going on?
I'm interviewing a microbial artist.
Ooh. Wow.
Her name is Michele.
For what purpose?
Michele Banks. She makes various kinds of art with microbes, bacteria, viruses, and she sells them. I've always wanted to interview her for my podcast, and I convinced ASM to send Chris Kondayan with his camera, and we're going to go to her house and watch her make things and talk to her about why she does this and so forth.
So does she depict microbes, or she uses microbes to make art, like with colorimetric strains?
She depicts them. She does watercolors. She makes scarves with microbes on them. I think some jewelry, kind of a diverse range of things. So Michele Banks.
Well, Abigail Saylors and I were on a panel in 2007 in Toronto that Chris Kondayan also taped with a group of artists in Toronto about using microbes in art. And it was another fascinating session.
So that's what Michele said, using the microbes themselves as part of the art. That's another thing that people do.
Yeah.
We talked about having a session at ASM General Meeting next year where artists come and present their stuff. I think that would be cool.
That's wonderful.
Microbial art. You could have a room with stalls, right? And you could even have them sell it. It would be very interesting.
Of course.
So, but we are going to have a session, actually, that was approved. We're going to have a session about people who make microbial art at the meeting. So I've always been interested in, I interviewed someone years ago who makes T-shirts and other things with viruses and bacteria on them. I'm always interested in, what got them started. So
we have a program here at Michigan run by the Organogenesis program called BioArt, where researchers are invited to submit their especially beautiful pictures from their experiments, and then they are made into note cards or posters or framed photos, or in some cases quilts, and then sold at the art fair, and the money then supports travel for trainees to go to meetings.
Wow.
So it's
And
kind
of course,
public
ASM has a program now, a competition of art on Petri dishes, right?
Oh, yeah.
Right.
Yeah. At Agar Art Competition, which I think the
Agar
deadline is September 3rd, right? And I will put a link to that in the show notes. So you can if you make an art out of an agar plate and submit a picture, and you can enter this contest. I'm one of the judges.
Ooh.
I have a block M, a blue block M made of E. coli on Bexgal.
Cool.
Uh-huh.
That's nice.
It would have been better if the E. coli was overexpressing the gene for indigo. I've seen that as well.
Michele, are you entering that in the contest?
Well, maybe I should. Thanks for reminding
me. If you go to microbeworld.org, on the left-hand side, there's a.
And you have an in with one of the judges.
Will that help or hurt?
I don't know. What's his allegiance to Big Blue?
Actually, one of the, if you like, here's a good point. The other judges, besides myself, Michele Banks, who is this artist, who she will be, Jamie Hensey, who Elio knows.
She's terrific. Great.
Dennis Bray, and and Sally Chisholm.
Wonderful.
Microbiologist and an artist. So after September 3rd, we'll start judging. Yes, so the deadline is September 3rd, 11:59 p.m. You just have to
it's cut off.
Take a picture
Eastern
of
Standard.
The Eastern Time, which I understand Ann Arbor is on as well.
That's right.
Okay, let's do a little bit of science. We have a snippet and a paper, and for the snippet, I'll turn it over to Elio.
Okay, well, today we're going to have a unusual situation that both papers being discussed or sets of papers being discussed are more or less on the same subject, namely intestinal microbiology. And this is going to be, I think, special because there is so much to the subject. And let me start out by saying, when you deal with something as complicated as the intestinal microbiome, you have to cherry pick something in order to talk about it. You can't talk about the whole thing. So of all, my job is to introduce you to a bug, a bacterium called Akkermansia muciniphila. Akkermansia muciniphila. It's a bug that you're going to hear more and more about because of all the various things it does. So in this cherry-picking exercise, selecting this bug makes a lot of sense. Why? Okay, first of all, it's an abundant member of the human intestinal flora. It degrades mucin. And since mucin is highly glycosylated protein which covers the intestinal epithelium, is very important in what goes on in the gut and what goes on in the bug with regards to bacteria. This is an important organism, and it does various and interesting things, and a lot is being said about it rather suddenly. So first of all, it was proposed as a new taxon in 2004 by some people in the Netherlands, Muriel Derian and colleagues. It's a gram-negative, strict anaerobe, non-motile, and it's a member of the planktomycete Verrucomicrobia chlamydia superphylum, for those of you who care. In other words, it's not a Bacteroides. That's an important thing to know. Okay, and it's been found in the GI tract of more than 90% of subjects analyzed. It constitutes between 1 and 4% of the fecal microbiota. Now, that doesn't sound like a lot, but think about it. We got, 10 to the 10th, 10 to the or so bacteria in there, so 1% of that is a lot of bacteria. 1 to 4%.
It's only one species, one genus and species that's representing it. So it really shows you, and there's between 700 and 1,000 to 5,000, depending on who you talk to, is in our gut of different microbes.
Why? Well, the question is, why has it been not noticed before? Why did it wait till 2004 to be discovered? And the answer, I don't really know, but I'm making this up. They isolated it by putting feces anaerobically in a medium where mucin was the sole carbon and nitrogen source. Now, it's entirely possible that although this technique, which used to be called enrichment techniques, which was started by the great school of microbial ecology in Holland, in Delft, that this particular setup was never used before. In other words, why would you look for something that can live only on mucin? But if you do that, out comes Akkermansia. Okay? So it's possible that that is the reason we haven't seen it before. Now, it's called Akkermansia in honor of Anton Akkermans, a Dutch microbiologist, and it's a recognition of his contributions to microbial ecology. Now, let me say a little bit more about Akkermansia. Generally, it has a large proportion of genes which encode secreted proteins. About 25% of its open reading frames are secreted proteins, and these include proteases, hydrolases, things that break down sialic acid, and so forth. So this suggests a specialization in things like mucus utilization, and possibly it can explain the gut, its participation in the gut environment. So it's a fair amount is known about it, and it does apparently some good things for us. Supposedly, in animal experiments and in correlation studies with humans, it has been implicated in reducing obesity, in decreasing type 2 diabetes. It correlates.
That's huge.
It's huge.
That's huge.
It is. It correlates, but get this, it's even better. It correlates with lower body weight and a healthier metabolic state. This is after calorie restriction. Okay? It is evidence that it really has to do with the gut health. In other words, you find fewer of those cells of Akkermansia in patients with ulcerative colitis or Crohn's disease. So both in the active disease and during remission. So healthy individuals have more of this bug, and when you lose this bug, there's a correlation at least with some serious diseases. Also, of all things, acute appendicitis. That blew my mind. So the fewer Akkermansia, the more the severity of acute appendicitis. So everything, I mean, wow, this is a super bug, right? And they correlate with the even type 1 diabetes and markers of inflammation.
Where can I buy it?
Where can you buy it? Now, wait. So it sounds like, it restores, it restores mucus layer thickness, and there is the, even though it breaks down the mucin, so I'm not sure how that works. And it counteracts also an effect which I didn't know about, namely an endotoxemia induced in obese mice. That is, if you inject lipopolysaccharide into obese mice, they die at a lower dose, and this counteracts it. So, perfect bug, right? A wonderful candidate for a probiotic. I mean, where can you buy it? Wait a minute. Hold on. Not so simple. It turns out that it also does something bad. It's correlated with inducing colorectal cancer.
Oops.
Okay. But that's specific to the fact that red meat has been found to correlate with the incidence of colorectal cancer. Why red meat? The emphasis here is on red. It's because in the myoglobin, a major constituent of red meat, which is what makes it red, it has heme. Heme is a risk factor. In rats, if you feed them a heme-rich diet, it damages the epithelium of the large intestine, induces it to proliferate and become cancerous. Heme, I've known that heme in tissues is it leads to inflammation. This is why when we have blood in our tissue, even a simple thing like a bruise, it takes forever to cure, right? You're black and blue forever, right? Mm-hmm. This is because heme is bad stuff. I mean, it's wonderful inside of red blood cells as part of hemoglobin, but outside it's bad news. It's a pharmacologically powerful compound.
So close the loop for us. How does this relate back to Akkermansia?
Aha. Okay. So it turns out that in bacteria like Akkermansia, they actually induce they loosen up the epithelium in such a way that things are absorbed better, including heme. So in the presence of bugs like Akkermansia, and this may have to do with the production of hydrogen sulfide, in the presence of that, it allows heme to get through the epithelial layer and into the cells. Heme is poorly absorbed otherwise. So if you were to use this bug as a probiotic, you gotta swear you'll never eat red meat again. Okay, so this is a Jekyll and Hyde bug. Most of the time, it is beneficial. It may lead to weight reduction, but it has this nasty way of being involved in the toxicity and carcinogenesis of heme. I suppose that one could probably engineer it in such a way that it doesn't do that nasty thing, but that may be part and parcel of what good it does. So this beneficial effects and this loosening of the membrane and allowing heme to get in may be tied together. I don't know. It remains to be seen.
All these effects that you noted are correlations, right? We don't have any causation evidence.
Well, in mice, you can you have a reasonable causation in mice.
But we would have to have it. We would have to have it in people before we could do anything. Does anyone trying to see if this would be a good probiotic despite its problems that you mentioned?
Well, I don't know. I mean, the answer is I don't know, but it seems to me that it's a candidate that should be taken, probably will be taken seriously. But on the other hand, as I say, we've been cherry picking. Mm-hmm. This is a big deal bug. Certainly, it's abundant. It's like Michael said, considering how many different kinds there are, to have one represent 1 to 4% of the total is a big deal. But maybe there are others. So this is a very complicated topic. I'm glad somebody else is doing it.
One of the things that I think with this microbe, because it took sophisticated enrichment to identify it in 2004, it never came out of the traditional, even back in the days when the Moors were working up the gut, and they were great anaerobic microbiologists. They were at Virginia Tech. Right. And so the Moors worked up a lot of it, and it didn't come out of their workup. And it was only unless they thought of using mucin as the only carbon and nitrogen source, it'd be hard to find, even if it's 1%. So the issue is, for a probiotic, you almost have to have a stable organism to lyophilize and then be able to put into, if you will, a pill or something that you can supply to an individual, or you have to make an anaerobic slurry and somehow ingest it without killing off the majority of them, because we don't really understand how to displace normal flora and then introduce the Akkermansia if you don't actually have it. And, I think that's what's so remarkable, is that it's been implicated in reducing obesity in type 2 diabetes. And the question is,
that's a good point.
If you see it in cancer, is it present at its normal 1 to 4%, or is it at 20%? Or, when you feed all this red meat, does it really go crazy, or it's just that whatever it's doing?
I don't think it does. No, I think that's not how it works. It doesn't respond to heme. It just allows the heme to get through
to feed the cancer cell.
But you're right. I mean, these are before you make something into a probiotic, you gotta, especially something that may be involved in disease, you have to think long and hard.
Well, and the paper that Michael's going to talk about today adds another layer of complexity, doesn't it?
Well, and that's why I picked this particular paper. It's because what Dr. Schloss's lab and his two phenomenal other co-authors on this paper did is really going to, drive home this studying it as a system concept that Elio's advocating for Akkermansia.
I didn't advocate anything.
Oh, I'm sorry. You're telling us to go look.
I don't want to get blamed for something I didn't say.
Yes.
Elio, are there other bacteria in our guts that remain to be discovered, or do we know them all?
You bet there are. Come on. I think Akkermansia is a wake-up call.
That's a loaded question, Vincent.
I like loaded questions.
I know.
So, but I bet there are other things we know very little about. I mean, it's all you have to do is to figure out a selective medium. Well, that may not even work. I mean, this idea of using a defined medium in the laboratory to fish out a bug with a certain physiological set of attributes is, as I say, it's old as the hills. It's 100 years old plus. And but it doesn't always work. So, but certainly I think there's this approach and there are many other approaches towards finding something odd. I doubt that we have a complete catalog of what's in our gut. In fact, I'm sure we don't.
All right. Thank you, Elio. And I will hand it to Michael. We've already had a good segue into your paper.
Yeah. The title of the paper that I'm going to discuss is Antibiotic-Induced Alterations of the Murine Gut Microbiota and Subsequent Effects on Colonization Resistance Against Clostridium difficile. And it's a three-author paper out of the University of Michigan, Go Blue, by Alexandria M. Schubert, Hamid Sinai, and Pat Schloss. And so the paper that Elio just introduced us to is a great segue into this particular manuscript. But I just want to start out with a term that they use a lot of in this paper, principally microbiota. And I want to give an operational definition of microbiota before I get to microbiome. And microbiota, you may or may not remember, was coined by Josh Lederberg. And the operational definition
Wait a minute, was it microbiota or microbiome that he called
Microbiota, Josh coined.
Was it?
And he emphasized the importance of the microbe inhabiting the human body in health and disease. And the operational definition that's out on the internet attributed to Josh is it's the ecological community of commensal, symbiotic, and pathogenic microbes that literally share our body space. Now, this is in contrast to some of the other definitions on the internet out there, and the one I'm going to quote is from allthingsgenomics.com, and they refer to the microbiome, refers to the entire habitat, including the microbes, their genomes, and the surrounding environmental conditions. So if you begin to parse words, are they synonyms or are they completely different things? Because, again, All Things Genomics goes on to say this definition is based on that of the biome, the biotic and abiotic factors of a given environment. And the author on All Things Genomics further argues that others in the field limit the definition of the microbiome to the collection of genes that are encoded by members of the microbiota.
By the way, we opened this up to discussion.
Oh, yes.
To readers of Small Things Considered. We had a poll, and the poll came out about half and half.
Yeah.
So there's no consensus.
No, there's no consensus, and it's really, I think, it's going to come down to the genetics of both the host and the microbes contributing in the ecological location that they occupy and the circumstance, what you're feeding them. It goes back to the paper that we just touched on. You can actually reduce obesity, you can lose weight, you can control type 2 diabetes. But if you eat too much red meat, all things are off because you could go down the path of colorectal cancer. So in that discussion, we know that the microbiota plays an integral role in preventing colonization by gut pathogens. We have this stable community of microbes in our gut, and Akkermansia just happens to represent a particular player. So in this work, what they have done is they show that this microbiota being able to protect the gut is crucial for protection against such pathogens as Clostridium difficile, which is the leading source of hospital-acquired infections in the U.S. today. And so well before this era of the explosion of papers elucidating the community of microbes on and in our bodies, microbiologists and physicians have appreciated that antibiotics are a major risk factor, or the consumption of antibiotics are a major risk factor for acquiring Clostridium difficile infections and the subsequent toxicity due to the manifestation of its toxin, due to the effect on the normal structure of the indigenous gut microbiota. So in this paper, we're going to learn how this group found that diverse antibiotic perturbations gave rise to altered communities that varied in their susceptibility to Clostridium difficile colonization. They found that multiple coexisting populations and not one specific population of bacteria conferred this resistance from, and they did it in a really ingenious way. The experiment that they used to elaborate the conclusion that's effectively manifest in their title is they would put mice on an antibiotic therapy regimen for a defined period of time. They would then take the mouse off of the antibiotics for 24 hours initially, and then they would challenge the mouse after allowing it to recover for a defined period. In the initial experiment, it was 24 hours. And then they challenged that mouse by giving it an oral inoculation of 1,000 C. diff spores. And they simply asked the question, plus and minus antibiotic, how many Clostridia difficile were in the animal's fecal material subsequent to the ingestion of the spores. And so colonization.
Let me ask you, this is lovely, of course, but does it represent something resembling the human disease, the artificial adding of spores?
Yes, because what happens in human disease is you get a blooming of C. diff, and when the C. diff blooms, it manifests the toxin. And so you get a large burst of C. diff at the end of the large intestine that then causes the pathology. And so they felt that by giving them in their model, giving them the spores, introducing the spores, because what we think happens with patients is the C. diff is ubiquitous in the hospital environment. The patient ingests the C. diff spores, and it doesn't take very many. The C. diff spores then germinate in the large intestine, and then they manifest the production of the toxin that then results in the disease. So you get this blooming of C. diff. Michele may have a better understanding. She's probably heard many of these graduate students give their talks, and they probably have addressed this. So if you want to comment.
I would comment one thing. What I really loved about the design is instead of trying to keep everything the same and get reproducibility, at exact same conditions, they actually embraced variation. So they gave a variety of different antibiotics, hoping that each would change the community in different ways. So then they ended up with, many, many mice that each had a different profile of microbial community. And then when they did the challenge, they could then ask, using sophisticated computational tools, what species correlate with protection, which correlate with susceptibility.
Yeah.
Okay.
And that's
beautifully.
The paper. Yeah, that's the paper.
That's the paper. We're done now. No. Thank you, Michele. The purpose of this investigation that they highlight is to expand the current knowledge of the effects of these perturbations by different antibiotics. And the antibiotics that they used was again, as Michele stated, they embraced difference. And so they had 2 antibiotics that would inhibit peptidoglycan synthesis. So the target is cell wall. And the 2 antibiotics were cephalosporin and a penicillin. They then had...
And more relevant is that they have different spectra, right?
Different spectra, exactly. And they used 7 antibiotics: ampicillin, cefoperazone, ciprofloxacin, which is a DNA gyrase inhibitor, clindamycin, which inhibits protein synthesis, metronidazole, which is a traditional antibiotic that you use to kill off anaerobes in a wholesale manner, and metronidazole destabilizes bacterial DNA by effectively alkylating it, and the net consequence is it effectively breaks the DNA apart. And they then used streptomycin, another protein synthetic inhibitor, different target, and then vancomycin. And each and every one of those antibiotics, as Elio said, has a different spectrum. The ampicillin targets gram-positives or gram-negatives. The vancomycin is traditionally thought to attack gram-positive bacteria. Clindamycin and metronidazole go after anaerobes, etc. And so what the really fancy part of their study, and as Michele said, is it's really pretty sophisticated, is they moved into this brave new world of bioinformatics where they used a methodology called random forest analysis that we'll get to. But in my shorthand, to keep that in the back of your minds as we go through their data, it's effectively ensemble learning language where they can compare and contrast these diverse sets of data that come out of the 16S sequencing that enables them to compare different situations and arrive at a conclusion. So we use 7 antibiotics. They stopped the antibiotics. They challenged the animal with 1,000 spores, and they observed reproducibly high levels of C. diff colonization in the mice treated with the peptidoglycan, cefoperazone, the generic anaerobe inhibitor, metronidazole, and then streptomycin. And they found in the stool, when they gave the animal 1,000 spores, they were able to see C. diff rates at a level of about 100 million per gram of fecal pellet. So that's pretty remarkable to see how quickly the C. diff bloomed in the gut of that animal. So they gave it, and 24 hours later they started collecting stool. And in the same case, they also then fractionated based on 16S ribosomal RNA sequencing. They then fractionated and reported out what was going on with the microbial community from the perspective of the OTUs, or what microbes were present in the fecal pellet of those animals. The curious thing about their first experiment was none of the mice that were given the DNA gyrase inhibitor, Cipro, either intraperitoneally or orally, were able to be colonized by the C. diff. And they also found some variation with vancomycin that would become important as they move down their path of trying to tease apart their data. So they start off with a really simple, straightforward experimental design, and as they take us through their data, they get more complicated. And what they principally did is they analyzed the molecular variants. And the molecular variants, and again, they're just using traditional p-values. And a p-value of 0.05 is significant, and anything greater than 0.05 was not significant. And so in their analysis of molecular variants, they found at a 91% confidence interval that Cipro could alter the community structure, and this then forced them to speculate that a higher dose of Cipro may be necessary to sufficiently perturb the community to a level to allow C. diff to overcome the colonization resistant. So as you begin to imagine what these authors did, they had lots and lots of mice and did lots and lots of sequencing of fecal pellets because they had 7 antibiotics times 2 because they had antibiotics and no antibiotics, and they analyzed before and after challenged by the spores in order to figure out who or what members of the community were rising or falling.
And can I just interject?
Sure.
When we say they, this is actually one PhD student and an undergrad who worked with her. So Alex Schubert is a PhD student who recently defended here at Michigan, and then Hamida was a undergrad who this was her first laboratory experience. So it was the two of them in the anaerobic chamber collecting lots of fecal pellets, preparing the DNA, and doing beautiful wet lab work, and then following it up with the computational biology. So I think that's really the power of this project.
So what they learned from that first set of experiments, where they're just treating the animals with 1 dose of antibiotics for a prescribed period of time, letting them recover for a day, they get some variation in their results. They now know that antibiotics perturb the gut microbiota, and that results in a non-overlapping community structure that yields significant variation and susceptibility to colonization when challenged by the spores. This conclusion forced them to make the next hypothesis, that titrating the dose of antibiotics that the mice received would result in smaller perturbations to the microbiota. Simply put, they expected a greater maintenance of resistance against C. diff colonization in these titrated treatment groups. So it's the same experimental design as before, but they lowered the concentrations of ceparazone, which is the beta-lactam, the protein synthetic inhibitor streptomycin, and then the lipophilic cell wall inhibitor vancomycin. And they selected these 3 antibiotics, again, going back to Elio's perception of the difference in spectrum of activity against these many types of bacteria. The cephalosporin will target the gram-negatives, the streptomycin will go after gram-positives, and the vancomycin will similarly go after gram-positives. And as expected, in all mice receiving the titrated doses, the beta-lactam, the ceparazome, C. diff colonization rates decreased significantly. Reducing the dose of streptomycin significantly reduced the colonization levels, and the vancomycin continued to cause them a bit of confusion as well in their studies. And what they learned is that what these 2 drugs did to the community structure is that titrating the dose of the ceparazone in the animal's drinking water resulted in these significant decreases in the relative abundance of these operational taxonomic units, or OTUs, associated with E. coli and a number of the other rarer OTUs that they had previously seen. They also observed increases in the relative abundance of OTUs associated with the family of the Porphyromonaceae. When they titrated the dose of streptomycin in the drinking water, this also resulted in significant changes in the relative abundance of these OTUs with the Porphyromonaceae family, the Allstipes genus, and the order of Bacteroideales. So again, now we go back to a metaphor that Elio used last week. So this is where we have the telephone book analogy. So you're giving me the telephone book of who's there. And now what we have to begin to use in this metaphor of telephone books, we now have to look at the telephone bill of who's talking to whom. And so what the results then suggested is that in the context in which specific members of the microbiota are found is important in determining the overall resistance to C. diff. And the example that they used in their discussion of their data is they specifically talk about the Bacteroides affiliated OTU and C. diff colonization was positive in streptomycin-treated mice, but it was negative in the beta-lactam or cephalosporin-treated mice. So that's why it's important to see who's talking to whom based on the issue of the drugs. In addition to the cephalosporin and streptomycin-treated mice, they had high levels of C. diff, although the former had significantly high levels of the E. coli-affiliated OTUs, which were absent in the streptomycin-treated mice. So again, you see how different drugs can alter the community differently. And so this illustrates how complicated these sorts of studies are done. And as Michele pointed out to us in the beginning, they really embraced difference. And they were hoping, I think, that that difference would allow them to make ultimately the conclusions that they were able to do.
Yeah, it was intentional that they wanted to develop this population of mice where each had been perturbed in a somewhat different way. And then they could, again, use this computational methods to glean a pattern. And the data are going to be gorgeous.
The next tumble they went down the path, again, keeping with this population of mice, is up to now we've only allowed the animals to recover for 24 hours before challenging them with spores. And again, the literature has suggested that communities can rebound and return to a healthy state in the context of being able to resist C. diff colonization. And this is really important in a clinical setting because when you do abdominal surgery, you want to prophylax the patient with an antibiotic. Anytime you open the abdomen, you're always afraid of nicking the bowel and letting the bacteria into that sterile space. So you want, antibiotics there. But what you don't want them is there long enough that the C. diff will get a foothold and take over. So that's where they next took, and this is the beauty of their translational aspect of their study. So in the next set of experiments, they ask how long are the animals at risk for increased colonization by C. diff if they expose and then challenge and look at population recovery. So they're going to look in the same sort of experiment by letting the animal on the antibiotic, take them off, and then they vary the time to recover. And what they used is an additional 5 days. And they, of course, measure the community by 16S. So just imagine these two individuals sequencing and picking stool and doing all the things they had to do in the anaerobic chamber. And so what they learned is amongst the metronidazole-treated mice, those with extended covering had a 2 million-fold reduction, 2 million-fold reduction in colonization. I mean, those data just blew me away. Talk about changes in risk. You allow them to recover for 5 days and then challenge, and you change the risk of colonization by 2 million. I mean, it's just phenomenal. In addition, 7 of the 14 mice that they treated that were given a longer recovery period had no detectable C. diff 24 hours after the challenge. So the community really responded. So again, going back to the phone bill metaphor, most notable amongst the 6 OTUs are what bacteria were talking in the community or coming up. These 6 OTUs were 2 of the OTUs that were affiliated with the members of the Barnsonelia and E. coli. And the relative abundance of Barnsonelia affiliated OTUs increased with extended recovery. So Barnsonelia would come up if they were allowed to recover longer. And the relative abundance of the E. coli, which was associated with C. diff colonization in those early experiments, decrease. Similarly, the metronidazole-treated mice, the ampicillin-treated mice that were allowed to recover for an additional 5 days before challenge also saw significant decreases in C. diff colonization. So from my selfish perspective of someone who worries about the built environment for protecting patients in the hospital, this drives home the need to keep that hospital clean, especially surgical recovery units, because you can wean them off of the antibiotics after they come off of surgery, but if the environment's dirty, they're likely going to get hit by C. diff. But if you just give them each additional day, really just confers that protection. So now we're going to go down the rabbit hole of the remarkable molecular biology analysis. The correlation analysis begins to address the fundamental question that many of you are probably all asking: what does this all mean? And how to make sense out of these ever-changing population structures. So they next did something really pretty clever. In order to identify the bacterial taxa that could be associated with resistance or susceptibility to C. diff across the 3 sets of experiments, they measured the correlation between the relative abundance of the community, or OTUs, on the day of inoculation with the level of C. diff colonization 24 hours later. So what they did is on the y-axis, they have this thing called the Spearman coefficient, where they compare and contrast the challenge with and without the spores, and they ask about OTU abundance, and they can actually measure just listing the families of bacteria on the x-axis. And so you can ask if it's a negative or positive Spearman coefficient, if it's going up or going down. And it, again, because this is all non-parametric measures, because the antibiotics don't normally change the distribution of the bugs in the gut because of the different spectrums of activities of the antibiotics that they're using. To illustrate what I mean by parametric and non-parametric distribution of bacteria, the next time you sneeze and you hit your monitor, just look at where your sneeze pattern is. You see a lot of the sneeze directly in front of you, but your sneeze goes everywhere. And so while the bacteria are concentrated in the middle of your screen, there are also microbes off to the side, above and below. And that's effectively what this antibiotic is effectively doing, is you're not getting normal distribution of the population based on the antibiotics killing everybody equally. And I guess the exception to that is clindamycin, which they curiously didn't comment on. And Michele, did they comment to you about the clindamycin control?
No, we talked on kind of a higher level about
this. So what did they learn? They learned that OTUs associated with providing resistance of C. diff outnumbered those associated with susceptibility. And the Porphyromonaceae affiliated OTUs were consistently associated with low levels of C. diff colonization. So that's the bug you want to have in your gut if you're challenged with C. diff. You want to have lots of Porphyromonaceae. And amongst the 3 proteobacters affiliated with significant positive association with a C. diff colonization, the ones you don't want to have in your gut subsequent to surgery is the E. coli and their associated OTUs associated with E. coli. And so their last set of experiments that they did.
Well, hold on. So figure 4, in addition to those points, what's really interesting is a couple of the families actually fell on both sides of the spectrum. In some conditions, the Lachnospiraceae, for example, were protective. In other conditions, they were they correlated with susceptibility, which goes to show that it's not the species per se, but who else is around.
Right.
So just a simple one-on-one correlation is not sufficient to make solid predictions about risk.
And that paradoxical observation suggested to them that colonization is a phenotype that is driven by multiple populations that act independently and possibly in concert to resist colonization.
Well, that's really the take-home lesson, isn't it, Michael?
Yes.
If you wade through this kind of forest of data, the thing that comes out is that it's not one set of bugs; it's a whole lot of them.
Oh, yes.
And they all participate in this phenomenon, right?
Yeah, and that's the last two figures of their manuscript, where I encourage everyone, because this is an open access journal, you can go and download this paper and look at the final two figures of the paper. And this is the random forest, the machine learning activity that they did. And this is what I found most fascinating about their work. This random forest machine home learning is you study the figures carefully, and what you take away from that combination of 15 different perturbations and the non-perturbed microbiota is that you generate distinct community profiles that are displayed in a range of susceptibilities to C. diff colonization. And this statistical learning tool, this random forest walking that they did, they were able to conclude that their findings demonstrated that colonization resistance was not driven by individual populations, but by a consortium of organisms. And intuitively, this makes sense to every community ecologist. It's the population that is altering the behavior. And in fact, their data goes beyond what others have done, where it was shown that Bartonella or the Lactospiraceae are partially protective against C. diff. And it was only because these two, this one graduate student and one undergrad, under Pat's careful guidance with them, figuring this wonderful experiments out, they had this large number of perturbations that they were able to demonstrate that a varied collection of populations was important for complete colonization resistance, leading them to summatively speculate that colonization resistance can be described as an emergent property of the microbiome, in which individual bacterial populations integrated into a community can contribute to the overall resistance to C. diff.
Yeah, if I could just add to that, the random forest regression data is shown in their figure 5 in this MBio paper, and it is absolutely beautiful. There are dozens of samples, but they each line up like soldiers on this regression plot. So even if you have no background in computational biology, you look at this and you're like, wow, it's very clear there's a pattern. And when Alex first saw this data, she thought, this is too good to be true. Have I fiddled with the parameters? So she manipulated parameter after parameter, tried different levels of sensitivity in the tool, and again and again and again got this really clear correlation. So it was just a fantastic experience in the lab.
Certainly, it's a very elegant piece of work, really. It's complicated, and I think we got to keep the take-home lesson in mind, because otherwise we get lost in details here.
Right.
If you're interested in how they did it, in their methods, there is a hyperlink that actually takes them to their code and the programming that they did, so you can actually see how they did the random forest thing. It's out on the internet. You just click on it, and it takes you to their workbook, if you will, of how they did it.
Michael, could you use this to design a probiotic to treat humans?
I think that's my take-home message, is that this random forest analysis you could actually see, and that's effectively where they take us at the end, is they say there are 6 sentinel species that you need in order to get us to that point. And I think it's only through this last set of analysis, the random forest regression model allowed them to describe community resistance and they're now in their discussion. They report on a paper, and they give the reference on a 6-species cocktail, including the Porphyromonas, the Lactospiraceae, the Lactobacillus, Coriobakteraceae, staph, Enterococcus, that successively resolved Clostridium difficile infection in mice. And this could actually supersede the need to do stool transplants. Right now, the FDA is having fits with qualifying stool donors to create a, if you will, ethical pharmaceutical for stool transplants to actually treat C. diff infections in humans. And so the probiotic here and the methodology that they present of dissecting this is really pretty remarkable. And I think one of the things that the NIH may wish to look at is to collect cohorts of patients that are on different antibiotics and then check their stool to figure out who's there and subjected to the sorts of analysis that Pat and his group did in order to figure out if the 6 cocktail that successfully resolved C. diff in mice could actually work in people to get away from the stool transplants.
Right. I mean, that's the question. Is there any reason to believe that it would be the same in mice and people?
So this group did do a study looking at the profiles of communities in the gut of patients with C. diff, and a number of the species that in their mouse experiments that we talked about today that correlate with protection are also turning up in humans. But to your point, Vincent, what they're doing now is taking human fecal samples, transplanting them into germ-free mice, and then doing the C. diff challenge to ask the same question. So they're trying to bring those two sets of data, closer together.
That's good.
Yep, very nice.
So the graduate student's going off to postdoc, but what is the undergrad going to do?
So the undergrad, Hamida, is now in pharmacy school at Wisconsin in Madison. But let me tell you a bit more about Alex Schubert. I've had the pleasure of knowing her since we were recruiting her. She was a bioinformatics major at Loyola, and she worked as a research assistant to an assistant professor at the time, Catherine Petoni, who herself is a computer science computational biologist. And because the lab was just 3 undergraduates, Catherine really gave them just a terrific experience. She encouraged them to drive their own projects, to write their papers, attend conferences, posters. And so Alex really gives Catherine a lot of credit for motivating her to continue her interest in applying computational methods to biological questions. So Alex, to her credit, has continued to kind of pay it forward. She's been involved not only in this great research, but is also active in a number of outreach programs for women in science at different levels, and also computational biologists in particular. So there's a program, for example, called Girls Who Code, which is to get high schoolers interested in computational biology. Another program called FEMS, which also is an outreach for high school students in math, engineering, and science. Another program that she's contributes to is called Science Delivered. This is a program that Olivia Mullins has started, and it's basically a mobile lab for kids in grades, elementary school kids, to get them excited about science. So Alex is really not only doing amazing science herself, but is also a really terrific citizen and is trying to encourage others to follow in her footsteps.
Lovely. Very nice.
Well, she did a fine piece of work here.
Yeah, and she also, I want to say, really felt fortunate to be training with Pat Schloss, who himself has formal training as an engineer. So he is
really?
Yep, that was his PhD, and then he did his postdoc with Joe Handelsman and learned the microbial ecology. But they're also fortunate to be working in a group we have here at Michigan called the Host Microbiome Initiative. So this is supported by our dean, that is bringing together the microbiologists, the clinical biologists, the microbial ecologists, the microbial pathogenesis people. They're providing infrastructure, including, for example, a lab that is just full of anaerobic chambers that is required for this kind of gut microbiology work. So it's really an exciting time here at Michigan, but of course across the country in understanding microbial ecology and how that's playing out in our bodies in health and disease.
Thank you, Michele.
You're welcome.
Thank you, Michael.
You're welcome.
We have time for a couple of emails. The first one is from Trudy, who writes, I'm sorry, I might have missed something, but the mechanism behind an effective vaccine is still unclear to me since infection with Francisella tularensis does not confer protection. How would a vaccine work? This has to do with our episode with Katie Bozio, who works on F. tularensis. So I asked Katie, and she replies, in general, any protection against tularemia is very dependent upon the dose of secondary infection, the route of secondary infection, and likely the subspecies of infection. Following infections with virulent subspecies, the evidence suggests that there is some minimal protection against low-dose secondary infection. In humans, this would be manifested as a very sick person, i.e. you might want to die, that had a delayed development of symptoms or someone with slightly attenuated symptoms. However, if the dose is modest to high, it's unlikely that the small memory response, one generated in the primary infection, will be effective. Moreover, infection via inhalation poses its own problems since the bacteria thrive within the pulmonary environment, while evading a suppressing inflammatory responses as a critical element if one wants to jumpstart adaptive immunity. Finally, if one is infected with a type B strain, the less virulent but still pretty nasty, there is some but not optimal protection against infection with fully virulent type A. All of this is supported by evidence from human trials using an attenuated vaccine strain LVS. This vaccine, which is no longer licensed in the U.S., was generated from type B tularensis and offered pretty good protection against low to moderate cutaneous infections. It also engendered some protection in one study, around 50 to 60%, against low doses of aerosolized Francisella. However, it was very poor at protecting against moderate, around greater than 1,000 doses of FT delivered by aerosol. Also, retrospective studies have indicated that people needed yearly boosters of LVS to protect against pneumonic tularemia. This supports the observation that long-lived immunity against pneumonic tularemia is difficult to achieve. We can recapitulate most of these findings in our animal models. So what do? My lab has generated some immunologic tools to really dissect how vaccines protect against low doses of FT, as well as how virulent FT modulates the innate and sequentially adaptive immune responses. By identifying what the host needs to survive on a molecular and antigen-specific level, we will be able to design a vaccine and vaccination strategy that gets the better of this bug. Hope that's helpful and glad to hear that people took an interest in this incredible bug. All right. So that's way more than we could have done. So thank you, Katie.
We got the expert to contribute.
That's right. The next one is from Julie, who writes, Hi, TWiMs. Thought you would like to know that you have saved many lives in Houston. When I listen to TWiM during my commute, I am not tempted to use my car as a weapon or a means to clean up the gene pool. I'm thinking too much. Road rage. Good. TWiM controls road rage. I teach microbiology at a community college and use TWiM for ideas that I can use in class, applications of the basic material we are covering, new information to update the textbook, etc. In a recent episode, you were talking about how important it is for students to do research to really understand science. We are incorporating that idea into several of our classes here. For the last 2 years, I have had my general micro students do research projects. The level of their questions has been amazing, and their excitement is infectious, pun intended. Our biotech students also do research projects in some classes, but also take advantage of a project lab that is open to students to try out ideas. It is equipped with equipment and some supplies for growing algae and bacteria, making biodiesel, prepping, scanning EM samples, making microbial fuel cells, and testing samples for bioremediation. I'm going to try to take this research approach to an introductory non-majors class. I'm trying it with honors students first by giving them choices of 6 different types of projects: bacterial pesticide degradation, DNA barcoding of algae and nutraceuticals. They have many different ways to go. They will get to work on projects that they chose, so I hope the buy-in will be there. You will notice how many of these projects are based in microbiology, great systems to use, little cost. Some of our projects are supported by NSF, others we just do because we can see the benefit for students. It was nice to hear that those of you at universities can see the benefit of what we are doing. it may be easier for us to do because we can focus on teaching. Thanks for all the mental stimulation. So Julie is a professor of biology at Lone Star College, Montgomery.
So all of you in the Houston area, go sign up for her classes.
Yeah,
they sound great.
I had a student in my virology class last semester who had not known any virology. She took it. She was turned on. She said, could I work in your class, in your lab for the summer? So she did, and she just finished. And she said, last week, this made virology real, actually working in the lab. Now I understand it. And that's so true. You know, it's abstract when you talk about it. But when you do it, you just run gels, do plaque assays, you really get it.
Yeah, and plus the biochemistry, the cell biology, it brings all those things to life.
Yeah, nothing like working in the lab.
And how.
Next one's from Robin, who writes about the anaerobic region of the mouth. This came up last time, is that right?
Yep.
The buccal cavity does have an anaerobic region, the gingivodental sulcus. That is why anaerobes are less likely to be found in aspiration pneumonias in endonculous patients.
What's that? What's
people without teeth. People without teeth.
They have no gingivodental sulci. Boy, proper brushing addresses the buccal and palatal lingual aspects of these sulci, while flossing takes care of the mesial and distal aspects.
All right, now she's showing off.
Now she's showing off. She's a dentist.
He's showing off. He's showing off. He's a former physician.
Robin, got it.
Wow. I couldn't even say most of it. Gingivodental sulcus, gingival dental, buccal, palatal, and lingual. Did I get those right, Michael?
Yep, you got those right.
And endonculous is without teeth.
Without teeth.
And mesial and distal?
Wow.
Near and far?
Near and far.
Well, now I know what flossing does. Thank you, Robin. Last one's from Dennis, who writes, Michael's discussion about tooth decay was very interesting and reminded me of a remarkable dental scientist and professor, Dr. Weston Price, who had discovered specific links between malnutrition and not only tooth decay, but also to malformed arches and consequential sleep apnea. His book, Nutrition and Physical Degeneration, was used in dental classes for 2 decades. A huge increase in tooth decay, sleep apnea, orthodontics necessary due to crowded arches, and more dental problems had ramped up by 1930. Turns out that a lot of it was due to a huge ramp in white flour and sugar, the flour having had all protein and micronutrients removed from it to make it purely white. So what does this have to do with microbiology? Stay with me, it's coming. Price and his wife traveled the world during summers for a decade and compared the teeth and arches of each group of people. He compared groups living mostly isolated from the Western diet and those who lived in a town with access to white flour and sugar. The differentiated groups lived close to each other, just isolated by what today would be short car rides. The differentiation became clear. Western diet, high cavity rates, non-Western diet, very low cavity rates. Anyway, the microbiome connection, he traveled in 1931 to a remote Swiss valley that was 9 days' walk from a town. He inspected the teeth of all of the kids in the school. He found only one cavity in 600 teeth, similar to but lower than the ratio of other non-Western diet children. But in this valley, everyone's teeth were covered with a green slime. They didn't brush their teeth, by the way. So I'm wondering if this is a clue to a microbiome that has teeth-protecting properties. Is anyone in the listening community interested in trying to track down what this microbiome was? You can say or post my email address as dennisrose40@gmail.com. My pick of the week is his book, which has experienced an increase in popularity due to its clear evidence and implication, $21 at Amazon. He provides a link to that. Keep up the great work. Gosh, the Twix on Wolbachia controlling male-female moth ratios and the possibility of a phage vaccine was all incredibly fascinating. Best to all, you are changing lives. Indeed, we saved them in Houston.
Mm-hmm. Well, I think he's talking about the wet spreaders. There's a group of bacteria that look like, if you're familiar with the family of myxobacteria, there's some that grow in the oral cavity that are wet spreaders that look like the slimes. And so the green slime may have been, Cytophaga. And so that may be the green slime that they're referring to.
More likely they're just polysaccharides made by some bug.
Yeah, that could be too. Probably strep mutans.
Well, that is TWiM 110. You can find it and all the others on iTunes or at microbeworld.org/TWiM. And if you have questions or comments, you want to tell us how we saved your life, please tell us. TWiM at twiv.tv.
Especially if we save your life.
If you didn't, we don't want to hear it.
Well, that won't.
Yeah, that's a joke.
Well, we could, around Halloween, we could have a seance, I suppose.
TWiM at twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you all.
Was anyone in the Schloss Lab interested that we were talking about this?
Oh,
paper?
yeah. No, they're very excited. And it turns out this is the second TWiM that we've done. So from the Schloss Lab. So the last one was another student on the impact of microbiome. It was our, what was the title? We gave it a clever title. The Little Shop of Horrors.
Yeah,
That's right.
Yeah, about colon cancer, I believe.
Yep. Colon cancer's Little Shop of
And
Horrors.
the microbiome.
Did you collect the check from Pat?
Yeah.
No.
She's doing the right thing. It's good stuff.
Yeah.
We just pick them and
just pick them.
And we're lucky to have the human microbiome initiative here too to support it.
yeah, that's great. Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure. What fun.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
You missed the pre-show, Michele. I was talking about, I have an old letter from Albert Sabin from 1982, and it's from the Medical University of South Carolina.
Wow.
I was there for a while.
Yeah, I got Sabin's old office when I first moved here.
Yeah, but now you're not in it any longer, right?
No, it's been renovated over and over. It's now a lab.
All right. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASM, the American Society for Microbiology, for their support of TWiM, and Chris Condayan and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.